let you know it's complicated, and we're going to go through some of the complications that we have. Gabriella did such a nice introduction, and so I'm not going to have to repeat a lot of this. You'll recognize the figure at the bottom. But what I always start with is just a reminder, a little bit of a reminder that when we talk about a dengue vaccine, we're really talking about 4 individual vaccines, because there's 4 individual viruses. And for the reasons that Gabriella discussed earlier, we really do need a vaccine that is safe and effective for each of the 4 individual serotypes. And that's been really difficult to achieve. We're hoping with a vaccine that will be able to induce long-lived immunity to each of the 4 serotypes, protective immunity at least against the more severe forms of the disease. We're not asking for sterilizing immunity, which is a really high bar. But what we have learned, for the reasons Gabriella mentioned, is that partial immunity to dengue is bad and can be problematic and lead to more severe disease with future infections. I think another important point that we had with dengue vaccine development is that we don't have a good animal model. So we can give a vaccine in animals and we can look for an immune response, but it's not necessarily predictive of protection. We saw that with early vaccine candidates that were essentially subunit protein vaccines of the envelope protein that looked really immunogenic in mice, but did not provide any really significant immunity in humans because mice identify different epitopes than humans do. And so we also don't have an animal model that replicates disease, so we can't test our vaccines to see if they're going to induce down the road more severe disease, so that's been a bit problematic. And we've always thought that neutralizing antibody is really indicative or predictive of protection, but I'm going to show you some of the results in the early clinical trials that really turned our head around and said we can't necessarily rely on neutralizing antibody. So I'm going to talk a little bit about early dengue vaccine work, and I'm at the Center for Immunization Research. I know other people are. I won't put you on the spot, but does anybody know who this is? This very famous vaccinologist. This is Albert Sabin. And before he worked on polio, he actually worked on dengue, and he identified dengue serotypes 1 and 2, which he called dengue serotype 1 was from, he isolated in Hawaii, and serotype 2 was New Guinea C. He had New Guinea A, B, C, and D, and it turned out that New Guinea C was dengue 2. So he, during World War II, as the Army often does, is looking for vaccines to protect soldiers. Mouse passaged these viruses, particularly the Hawaiian strain, which is dengue 1, in mouse brain. So he would inject mice in the brain and then recover the virus, passage it again, recover the virus, passage it again, until he finally found one that was attenuated. And he noted that after 7 to 10 passages, the illness was modified. It wasn't asymptomatic illness, I'll tell you that when you look at his early papers. There still was fever, there still was some rash, but certainly modified. It did not move forward after, with Albert Sabin after World War II, but other investigators did take it forward. And Japanese scientists were doing the same work, and there was a large outbreak in Osaka in 1943, and they again passaged the virus in mouse brain. They didn't know what strain of virus it was at the time. But what's interesting and why I'm presenting this is a paper that really shows you can have very long-lived neutralizing antibody to dengue years and years after inoculation or infection. So 40 years later, they brought people back who were in these original experiments, and they measured neutralizing antibody, and they showed that the virus they were infected with was the mouse-modified dengue 1 virus. And 40 years later, they still had what we would consider to be very good titers of virus. And this, again, is something where we think live attenuated vaccines can induce long-lived, we hope, protective immunity. So the current vaccines that I'm going to talk about today are all live attenuated vaccines. They're called tetravalent, meaning they have 4 components. At the end of my talk, you can decide whether they really are tetravalent in action and not just in name, but we'll talk about that in a little bit. Some of the reasons we like live attenuated vaccines is because the vaccine presents your body with a modified pathogen that doesn't make you sick, but your immune system sees that pathogen, or sees the vaccine as it would the pathogen. So you get a very good Th1, Th2 immune response, you get good CD8 T cells response, you get good humoral immunity. They're generally less expensive to produce, but they can be very difficult to develop, which we'll talk about in a minute. You get long-lived protection, think about measles vaccine, think about yellow fever vaccine, but they are contraindicated in certain populations, so they're certainly not for everybody. But I think the most important point I want you all to remember is that for a live attenuated vaccine to work, that vaccine has to infect and it has to replicate, because your body is the vaccine manufacturing plant for that vaccine. We give such a small amount as an inoculation, we're betting on that virus to replicate within the host, to be presented in the immune context that the pathogen would, so that you get a very good, broad, long-lived response. So these are the 3 vaccines that I'm going to talk to you about. Dengue, most of you may have heard about Dengue. I think it really is, I always say this, we learned more about dengue, I think, from the Dengue vaccine study than we ever learned about all of the studies we've done in mouse and other studies. We learned a tremendous amount. And there's certain things that